All right, so today we're going to talk about Hardy-Weinberg equilibrium, and actually the notes for today are going to be kind of short, and then you will have a worksheet to complete that are examples of this. The worksheet should only take between 20 and 30 minutes, even with like starting slow because you don't understand the equations right away. So take your time, but make sure that you get started on it before my final office hours so that you can ask questions if you need to. Um, and then use the rest of your class time today to work on your outlines again. All right, so the Hardy-Weinberg equilibrium. We had two scientists, Hardy and Weinberg, obviously, who came up with this theory, and it provides a framework for understanding how populations evolve. So we have a couple equations. The basic equation, the one that is sometimes referred to as the Hardy-Weinberg equation, is p squared plus 2pq plus q squared equals 1. There's also, p plus q equals 1. And all this is telling us is that the dominant allele is going to be p, the recessive allele is going to be q, and that together our gene pool will have 100% of all alleles will be either that dominant or that recessive. Now, obviously, this is only going to work on things that have two versions, two alleles. We've talked about some things where there are more than two, so then it would be more complicated, but we are not going to worry about those in the population genetics with Hardy-Weinberg. We're just going to worry about our straight two different alleles for a given trait scenarios. And then p squared plus 2pq plus q squared, this is basically our Punnett square. So if you've got your p and your q, here is p squared, here is q squared, and here is pq and pq. So if you had a dihybrid cross, you end up with p squared, q squared, 2pq. All right, so now I'm going to talk about in which populations this would work. Hardy-Weinberg equilibrium describes populations that are not evolving, populations that stay the same, not evolving, and the genotype frequencies would stay the same if 5 conditions are met. And so these 5 conditions would have to be in place in order for Hardy-Weinberg to actually describe a population. It would have to be a very large population, so no genetic drift. It would have to have no emigration or immigration, so no people leaving the population, no individuals moving into the population. And I said people, but this could be animals or plants or whatever. No mutations, so no new alleles being added to the gene pool. Random mating, so that means no sexual selection, no selecting for the bird with the reddest feathers, none of that. And then no natural selection, so all traits should aid equally in survival. There should be no selective pressures on any of the traits. So are natural populations even in Hardy-Weinberg equilibrium? Well, no. Immigration and emigration happen, like individuals move into and out of a population all the time. Errors in replication happen at a frequent rate-ish. It depends on what genes we're talking about. So it's not like super common, but it does happen at a low constant rate. And sexual selection happens in a lot of species. So there's not really any natural populations that would be considered in Hardy-Weinberg equilibrium. However, it is useful because predicting the genotype frequency can help us understand more about the population. So predicted genotype frequencies and how those compare with actual frequencies can tell us something about the population. It's used for traits in simple dominant recessive systems, and you have to know the frequency of recessive homozygotes. If you know the frequency of recessive homozygotes, you're good. You can go from that one number to everything else. And I'm going to show you by the end of this lesson how that would work. So here's the same thing I drew over on the last slide. Here is what our variables are. So q squared, or we'll start with p squared since it's at the top, is the frequency of like the dominant homozygote, so big T, big T. Q squared here is the frequency of the homozygous recessive, little t in this case. And then, of course, our heterozygote is our 2pq. All right, so just to get us started with something kind of basic to kind of go back to that term gene pool. In the following population, what would be the allelic frequency for the dominant allele? We have 20 homozygous recessives. So let's go ahead and use, let's think of a good letter. Let's use Q. We'll just use big Q and little q. So our 20 homozygous recessives are little q, little q. We have 320 homozygous dominants, so that's big Q, and 160 heterozygotes, big Q, little q. So all we need to figure out if we want to know the allelic frequency is we need to figure out our total number of alleles and our total number of alleles of each type. So if we have 20 individuals that are little q, that means for little q, we have 40 copies of that allele in these individuals, okay? We have 20 individuals, they each have 2 copies, so 20 times 2 is 40. The same thing here, we can do that with big Q. So we have big Q, we have 320 times 2, which is 640. And again, that's only here. Now we have these heterozygotes. The heterozygotes are a little bit more complicated because they don't have 2 copies of each allele, they have 1 copy of each. So we take that number of heterozygotes, 160, we add it to there, and that would be for your big Q, and then we add it to here. So little q's, we have 200, big Q's, we have 800. So total, we would have 1,000 alleles in the gene pool. So our little q would be 200 divided by 1,000, which would equal 0.2, or you could say 20%. And then our big Q, that could be P if we're going to the Hardy-Weinberg letters. Remember, I'm just using the letters that represent the like dominant and recessive genes. So this could be 800, or this would be 800 divided by 1,000, which equals 0.8 or 80%. So now our question was asking the allelic frequency for the dominant allele. So the one that is dominant is our capital letter here. So our answer would be either 0.8 or 80%. So that would be our answer. And unless I specify, you could give me either one. I'm not going to count wrong if you put 80% instead of 0.8. All right, so now we have a little bit more complicated. So if 16% of the persons in a population show a recessive trait, what is the allelic frequency for the dominant allele? Now, this is key. They show a recessive trait. So that means they're homozygous recessive. So our 16% equals our Q squared from our equation. So Q squared equals 16% or 0.16, okay? Now remember, our equations are P squared plus 2PQ plus Q squared equals 1. Now, right now, I only have Q squared, so I got to figure out the rest. But I also have the equation that P plus Q equals 1. So what I can do is I can take this and I can find Q. So I do that simply by taking the square root. So the square root of 0.16 is 0.4. So Q is going to equal 0.4. So if I put that into my equation here, I have P plus 0.4 equals 1. And so I can subtract 0.4 from both sides of the equation. And so P equals 0.6. So now I know P, in addition to knowing my Q and my Q squared, right? So now what I can do is I can find P squared. P squared is going to be 0.6 times 0.6, which equals 0.36. And that would be 36% of individuals. Then I would have, so my equation, remember, is P squared plus 2PQ plus Q squared equals 1. Now, the other thing I can do is I can just subtract, or I can check my work. So I can just say, okay, I know that I have 16% Q squared, 36% P squared. So 16 plus 36 is 52. So I have to have 48% in this 2PQ category. Or instead of doing that, what I can do is I can actually figure it out. So I know that I've got my p squared is 0.36 plus, and my 2pq is just multiplying 0.6 times 0.4. And then, of course, times 2. Can't forget that on the outside there. Equals 1. And, of course, we can double check at the end, make sure it equals 1, right? That's how we check our work. So 0.6 times 0.4 is going to be 0.24, but then times 2 is 0.48. So 0.36 plus 0.48 plus 0.16, and that should all equal to 1. And if you do the math, it does. So now, what is my question? Because I know all of my different variables here, so I've got all that figured out. So what is the question asking me? The question is asking me what is the allelic frequency for the dominant allele. So it's just asking me for Q. It's just asking me for that allelic, or for P, sorry, for the allelic frequency of that dominant allele. So my allelic frequency for P is 0.6. So that's our answer. So you didn't even have to do all this. I could have also asked you, though, how many individuals are homozygous dominant? And then you should be able to tell me, okay, homozygous dominant is 0.36 or 36%. I could ask you, I could give you just this information here, just that, and I could ask you what percent of the population are heterozygotes. So from just that one bit of information, you can find everything else. So I worked it all out to kind of show you how we can find all that information, even though the only part I needed for the answer was this part over here that was way at the beginning. All right, I hope that that makes sense. I'm going to go ahead and end this video. You guys will have practice problems online. You should do those. And I will be in office hours. We can do the whiteboard thing so I can draw on it and go through some more example problems if you're still confused. This tends to be a little bit of a confusing topic, but once you get it, it usually kind of clicks and then it's not too bad. So if you're not getting it, don't ignore it. Like, this is important. You will see this on, some sort of assignment, maybe a test in the near future. All right, that's it for this one. So your HW practice or Hardy-Weinberg practice will be a separate worksheet. So you don't have to worry about turning in any questions from the lecture.